Hello students, this is your science teacher Mr. Pierce with part 2 of my videos on how to use a microscope. Please follow along with the PowerPoint video to help you become familiar with how to care for a microscope, the different parts which you should already know from filling out the card last week, as well as focusing the microscope. Let's get started. When caring for your microscope and transporting it from place to place, you always carry the microscope with 2 hands. From becoming familiar with the different parts of the microscope, you should already know that this part is called the arm of the microscope. The lower part of the microscope is called the base. When carrying the microscope, one hand is always holding the arm, while the other hand is always under the base. You may practice lifting your microscope from the counter now. To clean your microscope, you only use lens paper. If you ever try to use anything other than lens paper, you run the risk of scratching the lenses of the microscope. When using your microscope on trying to focus with the focus knobs, you may find that the knobs will sometimes stop. Once the knobs have stopped, you cease turning them. If you continue to turn them, you will break the gears. Please make sure that when the knobs stop, you do not force them to turn anymore. You stop turning. When you are using the microscope, when you are done using the microscope, you always cover it back up. Also, when using the microscopes at your counter, please try to keep your counter space clear. Obviously, you will be having a lab sometimes. It is appropriate to have a lab in the location, but try and keep other objects clear from your workstation, as well as the cord. So, this is question number 1 that you are to answer on notebook paper. Please remember to write your answer in a complete sentence. What part of the microscope do you hold while carrying it? Please take time to answer now. If you need to pause the video, do so. Your next question. It is a true-false question. Remember to answer in a complete sentence. If the object does not focus, then you keep forcing the knobs until it does focus. You may want to pause the video to answer. Next, when finding the magnification of your microscope, you will be familiar with that the eyepiece has a magnification of 10x or 10 times. Our microscopes in our class follow that rule. Some microscopes that you use outside of class may have different magnifications. There are 3 objective lenses. Our 3 objective lenses have magnification of 4 times, 10 times, as well as 40 times. These magnifications are standard for most compound light microscopes. There are some microscopes that have a magnification of 100x, 100x, or even sometimes greater than that. We call those oil immersion slides, or oil immersion objectives. To find the total magnification, you take the eyepiece and you multiply it by the objective lens that you are looking through. For example, if you're using a 4x lens, objective lens, what is the total magnification? You should do the math. 4x times 10 is a total magnification of 40x. Question number 3 for you to answer on your notebook paper. If the eyepiece is magnified 10x and the objective is magnified 10x. What is the total magnification? You may pause the video if you find it necessary. Moving on. Our light microscopes are capable of magnifying several things that you cannot see with the naked eye. However, there are more powerful microscopes out there. They cost a lot of money. It is not in our science budget to purchase one. We call these electron microscopes. They're capable of seeing things like lactobacillus and yeast. Our microscope, well, this is showing you yeast in both the light microscope in the middle image as well as yeast with the electron microscope in the right image. This is a diagram showing you the capabilities of what you are able to see with what tools. With the standard naked eye, humans are capable of seeing things much larger than a human, but also all the way to about the size of hair. Your light microscopes, which you will be using in the classroom, are capable of seeing things in a range of smaller than a centimeter to that of almost 100 nanometers. You are able to see micrometers. That is what the scale is on top showing you. Looks like the U with a tail. That is a micrometer. So you are capable of seeing things like ants under the microscope, hair, individual cells, bacteria. You are not able to see viruses or things that are smaller than a virus. Standard electron microscopes are capable of seeing things in a range that is slightly greater than 10 micrometers, all the way down to almost atomic size. I'm going to go up here now to allow you to see that the electron microscope is not capable of seeing individual atoms, nor is it able to see electron orbitals. However, electron microscopes are capable of seeing viruses, DNA, small molecules, but not yet an individual atom. Which leads us to our next question. What can a compound light microscope not see? Please answer in a complete sentence. These questions will continue to get harder and be less multiple choice like. You may pause the question if you need the time. Question number 5. What is just out of the range of electron microscope to make it visible? You may want to go back in the video to look at that diagram again. Please answer in a complete sentence. This has been part 2 of your introduction to microscopes. I am Mr. Pierce, reporting from my basement. Goodbye.